会要到3点半，待会就直接开啊。到时候你直接切换视频。
切换视频的话，不是它这个界面会关掉，
得再得重新共享一下。
重新共享那意思，是叫共享
对。对。
那个吗？
点这个就会结束。
你上午也在这里。
有那个Zoom吗？Zoom好了已经，今天好了，没那么快啊。Zoom
提前开了，
要不看这
应该不。
Okay, that's fine. So we go.
Thank you, man. I should sit in the my body. Okay, Bobby.
Recording in progress.
Yes, yes. So I said in front. I will make story. 开始了吗？还需要讲还是不用讲了？那就点开始了，顾老师。
Hello, everyone. I will give a lecture about virality and manuscript. Sorry, I provide a lecture video. Okay.
Good afternoon, everyone. Welcome to my presentation on virality and kind analysis. My name is Huang Shaohua, and I will explore a fundamental property of our universe. Virality It's a concept that scales from the vastness of space right down to the molecules that build life itself. Here is a brief look at our agenda for today. We will begin by exploring the mystery of chirality, observing its presence all around us in the natural world. Then we will spend a significant portion of our time discussing the critical relationship between chirality and health. Finally, we will introduce kind drugs and kind analysis. So let's begin with the mystery of chirality. The concept of chirality is handedness, and once you start to look for it, you will see it everywhere. We see it in a microscopic world. For instance, in the growth patterns of climbing plants. Notice how this cucumber tends to coil right-handed helix, while the wisteria often, though not exclusively, prefers a left-handed twist. And some climbing plants, for example, vines, show right-handed growth pattern, while others display left-handed growth pattern. This asymmetry extends to the beautiful intricate structures of flowers. The petals themselves can twist in defined left or right-handed helices. A clear demonstration of botanical chirality. In the animal kingdom, it's just as prevalent. The coiling of a snail shell is a classic textbook example. Most shells are dextral or right-handed. However, rare sinistral or left-handed shells occur. This is not a random occurrence. This handedness genetically determined at the earliest stages of embryonic development. Even bilateral symmetry of animal body, left side being a mirror image of right, is a form of microscopic chirality. But this concept isn't just for large-scale objects. It scales all the way down. Chirality exists at the subatomic level. For example, in the spin of an electron, which can be up or down relative to its motion. And this is where chirality becomes critically important for life. As we know it, the fundamental building blocks of life are themselves chiral. The proteins in your body are made exclusively of L amino acids. The DNA that encodes your very being is a D-sugar-based right-handed double helix. Life is built on a principle of homochirality, to use only one hand for its essential components. So scientists are now even building a synthetic mini-image LDNA enabled to study why nature made this choice. We, as humans, have even begun to mimic nature's chirality, creating synthetic mesoporous materials with chiral structures, and designing a stunning chiral architecture like this spiral staircase. This pattern of handedness extends to the largest scales imaginable, from the spiral of ocean currents and hurricanes driven by the Coriolis effect, to the magnificent spiral arms of galaxies. So as this summary image beautifully illustrates, from the subatomic neutrino to the molecule, to the biological, and all the way to the cosmic chirality, is a fundamental and universal organizing principle. Now that we have seen where chirality exists, let's formally define what it is. The word chirality comes from the Greek word chair, meaning hand. This is the perfect analogy. As you can see, your left hand and your right hand are perfect mini image of each other, but they are not superimposable. You cannot place one perfectly on top of the other so that all features align. This fundamental property, being non-superimposable on one's mini image, is the definition of chirality. The exact same principle applies to molecules. A chiral molecule, like the amino acid shown here, typically has a chiral center, a carbon atom bonded to 4 different groups. This arrangement results in two different molecules called enantiomers. There are a left-handed and a right-handed version. They are mini images, but they are not the same molecule. This groundbreaking concept was discovered in 1848 by the brilliant Louis Pasteur. He was meticulously studying tartaric acid crystals, a byproduct of wine fermentation. He noticed under the microscope that there were 2 distinct types of crystals, and they first made them many images of each other in what is described as the most beautiful experiment in chemical history. Pasteur used a pair of tweezers to painstakingly separate the two crystal types. He then dissolved each type in water. He found that a solution of one type rotated the plain polarized light to the right, and the other rotated it to the left. A 50-50 mixture of the two, which we call a chemical mixture, had no effect on light at all. This was the birth of stereochemistry. Pasteur's discovery raises a profound unanswered question. Why? Why did life on Earth select L amino acids and D sugars? Why not the other way around? This is the mystery of homochirality. There are several competing theories. The life-based theory once suggested that life arose from the chemical primordial soup, and then selected one enantiomer for its structures. However, this has been challenged by findings that self-replication might not even be possible without pre-existing homochirality. This leads us to long life-based theory. Perhaps there was a random event, a 50-50 cosmic coin flip that just happened to land on L for amino acids, and life followed, or the deterministic theory suggests that some kind of physical force, like circularly polarized light from a neutron star, or even a fundamental weak nuclear force, interacted with molecules interstellar dust clouds, preferentially destroying one enantiomer and leaving an excess of the other. This kind of bias was then delivered to Earth by a meteorite seeding the template for life. This might all seem like abstract theory, but the implications of chirality for our health are immediate, profound, and sometimes tragic. This brings us to the thalidomide incident in the late 1950s and early 1960s. A drug called thalidomide was sold as a racemic mixture, both enantiomers combined as a over-the-counter sedative. It was particularly effective for treating morning sickness in pregnant women. Here is the problem. The molecule R-thalidomide, the right-handed version, was indeed an effective and safe sedative, but its mini-image S-form, S-thalidomide, was a potent teratogen. It caused catastrophic birth defects, resulting in over 10,000 children born with severe deformities such as phacomelia. This avoidable tragedy taught the world a painful lesson in the biological system. The two enantiomers of a drug are not just similar, they are two different drugs. And thalidomide is not an isolated case. There are many other kind of drugs similar to thalidomide, such as ketamine, and some butyl, propoxyphene. Why? Because our body's receptors are themselves kind. They are the logs. Only one specific kind key, the enantiomer, will fit and produce the desired defect. The other key might not fit at all, or it might fit at different unintended log, leading to disaster. This crucial principle extends beyond medicine into our food and our environment. Even your sense of smell is a kind of detect. For example, covering is the molecule that gives spearmint its characteristic flavor and aroma. Its perfect mini image is cover smells completely different, like calaver, a spice in rye bread. Same atoms, same collections, just different handedness and totally different biological response. Similarly, as anemone is the bright, fresh scent of nemours. Its enantiomer anemone is the familiar smell of oranges. Chiral also matters in food safety and processing. As we established, all natural proteins in our body coating air amino acids. However, high temperature processing like gardening can cause a chemical reaction called racemization. This converts some of the natural air amino acids into unnatural D amino acids. The long-term health implications of consuming these racemized proteins are still an active and important area of research. This concern is paramount in the environment. Many modern pesticides are chiral molecules. Let's look at triazole fungicides. The parent compound triadimerphine is metabolized in the soil and in the plant into triadimelol. This creates two kinase centers resulting in 4 different isomers. These isomers have been shown to have different toxicities and critically different environmental persistence. One isomer might break down in a few days. Disappear quickly, while its mirror image might persist in the soil for months, posing a long-term risk. This slide shows 2 more stark examples for the pyrethroid insecticide on the left. On the left, the 1R. On the right, the 1R 3R isomer is the potent insecticide, while the 1S 3S isomer is toxic to humans. For the herbicide on the right, the S form kills weeds. S form kills weeds, but the R form is harmful to people. That is why regulatory agencies worldwide, like the FDA and EPA, now increasingly demand initial drugs and pesticides. The goal is market only the single active self isomer, reducing the patient's metabolic burden and total amount of chemical released into the environment. Now, I have said our bodies are built from amino acids. That is true, but it's not the whole story. We are now discovering that small but significant amounts of D amino acids, particularly the aspartate and D-serine, exist naturally in our bodies. They are found in very slow turnover tissues like tooth enamel, bone, and the crystalline proteins in the lenses of our eyes. Their accumulation appears to be linked to the aging process. As the body's natural amino acids slowly racemized over decades. This accumulation is correlated with age-related diseases like Alzheimer's and cataracts. So where do they come from? Some are endogenous, as I just described, that slow racemization over time, but many are exogenous, meaning we consume them. Fermented foods like yogurt, cheese, soy sauce, and wine, as well as various processed foods, are all long sources of D amino acids. This is fascinating and emerging field of nutrition and medical science. Given all of this, let's briefly revisit the kind of drugs to categorize them. When a pharmaceutical company develops a new kind of drug, its properties generally fall into one of these types. In some rare cases, the enantiomers have the same pharmacological activity and similar potence. The anesthetic bupivacaine is an example. Here, a racemic mixture may be acceptable, though the S form also has vasoconstrictor effects, making it a slightly better drug on its own. More commonly, the enantiomers have the same type of activity, but different potence. The classic example is ibuprofen. The S form is the potent anti-inflammatory. The R form is much weaker. Interestingly, our body has an enzyme that can slowly convert the inactive R form into the active S form, which is why a racemic pill is still effective. And finally, as we have seen, the most critical cases are those where enantiomers have opposite or entirely different pharmacological activities. S-ketamine with R-ketamine. This must be separated for safe use. Okay, this slide brings us to our final topic, kind analytic methods. If you have a mixture, how can you tell the enantiomers apart? They have identical melting points, boiling points, and a standard spectrum. It's incredibly difficult. difficult. The gold standard in modern chemistry is chromatography. This is a powerful separation technique. The most common direct method is to use a stationary phase. This is a column for an HPLC or GC instrument that is itself called with a molecule. As the racemic mixture passes through, one enantiomer will shake hands or interact more strongly with this phase, causing it to travel through the column more slowly. The other enantiomer will pass through more quickly. The result, as you can see in this chromatogram, is a beautiful separation of the two enantiomers into two distinct peaks, allowing us to quantify them. We can also use spectroscopy techniques like circular dicolorism, measure how the two enantiomers absorb left and right circularly polarized light differently. Or, as shown here, we can use nuclear magnetic resonance. By adding a shifter reagent to SAMP, we can make the two enantiomers, which normally appear as one signal, show up as two different distinct separate peaks. Other methods such as biochemical assays and electrochemical analysis are also powerful tools in the analytic chemist's toolkit. So, conclude this lecture, we have traveled from the spirals of galaxies to the core of our own DNA. We have seen that chirality is a fundamental, universal property of matter. We defined it using the hand analogy and saw its humble discovery in Pasteur's tetrate crystals, and most critically, we have understood its profound and wide importance in our health, our medicine, and our environment. Optimized by the thalidomide tragedy, the ability to distinguish and separate pure enantiomers, analysis, and preparation is not just an academic curiosity. It is one of the most important and challenging cornerstones of modern chemistry, pharmacology, and environmental safety. Before you think about these questions, I would like you to just take a moment to reflect on these key points. Okay, thank you very much for your attention.
This is popularization strategy. Okay, and then we
Good afternoon, everyone. Welcome to my presentation on chirality and
now next is Dr. Wu Dan Dan. Dr. Hong Bili will introduce the ALMA analytical method.
Hello everyone, I'm Huan Bilin. Today I will introduce about.
Can you use mine?
Hello.
Okay, not good.
Okay.
Two slide, but still your
Yeah, your voice.
focus sound is too low.
गाना थोड़ा गा रही है
That's a सुंदर sound. Yeah, it's working.
Good afternoon everyone. Today I will introduce about NMR in analysis. I will introduce from the following six aspects. I have.
Can you make the slideshow?
Like shift to the presenter mode. The cursor please. Presenter mode. Yeah.
आज ज़्यादा समझ रही है
Okay.
I have recorded audio before, so now I will play it to share about this report. Chirality is a fundamental and the universal property of nature, showing across scales from subatomic to cosmic levels. The two most notable examples are chiral amino acids and the nucleic acids. In nature, amino acids exist in the L form, while sugars are D form in the nucleic acid. Chiral molecules are mirror image and cannot be superimposed. Each molecule features a central atom bound to four different substituents. There are also some actual chiral compounds. The enantiomers have similar physical properties, thermodynamic and chemical properties, but they have different effects in biological systems, where the intrinsic homochirality has profound consequences for pharmaceutical chemistry. The specific structure activity relationships may be required for effect. For example, enzymes and the nucleic acid. The physiochemical and the biochemical properties of racemic mixtures and the individual stereoisomers can differ. In pharmaceuticals, one enantiomer might be
这个共享
有啊，在共享，
这个杂志他就看不见
谁共享的？
这个共享
就是哎，在共享了呀。
刚刚把车上
嗯，不是的，
别人那个哪个
共享了的呀。
展示一下共享
不要选了没？不要选。
Escape करें ना.
他说还可以帮忙
Do escape.
Share on Zoom?
Escape करें ना, escape करेंगे तो ऊपर options आ जाएंगे.
क्या?
इसको normal PPT mode पे लेके जाए ना.
नहीं आ रहा है, ये मेरे पास आ रहा है यहाँ पे.
अच्छा.
Professor Pro said she cannot see the screen.
So
Yeah.
what is written here? Maybe we can ask.
Can you ask someone
Yeah.
online?
Yeah.
Yeah.
赵老师还没看到，
因为他问过其他人，
对对，问一下其他人的，
他在聊天框里
问一下其
聊天。
他人的看到。
Have sufficient time for one.
OK，还有吗？
人国
It's a biology teacher student about parallel now.
maybe it's here.
But it's a subject.
It's here, sure.
Is of
Yeah, this. No, this is.
Oh,
No, this is okay.
share.
No. Okay.
Three.
No, this is okay.
That is this three.
This is all. This is okay.
点一下这个，然后
Again, no.
In our body have some amino acids from plant, right? So we have microbiota in our intestine.
你再看一下那
Here,
个
someone have tested.
Those lines are still up to the.
Yes, they are
Yes.
online.
It's
Online
okay.
are okay.
This is.
Online.
This method so that the bios.
就像刚刚那个，就是刚刚那个，
他们那边可以看到这个，那就继续讲。
那这是那刚才那个是没有 share 类的。
be therapeutic, while the other could be inactive in the harmful.
Your mind is.
When a new chiral molecule was created, it's necessary to determine its absolute configuration or chirality, even the entire process of drug production. This is very important for drug safety control. However, how to distinguish two molecules with the same atoms? How to assign their chirality? From the historical optical polarimeter and to modern instruments in the supramolecular process, this research field is developing with the purpose of finding techniques of more general applicability and reducing the time and the cost. Today, widely employed techniques include chromatography, NMR, optical spectroscopy, mass, and fluorescence. The difference in the principles of these measures allow their combined use and have been applied in drug development and stereochemical research. Here, we mainly focus on the application of NMR in chiral analysis. NMR cannot differentiate to enantiomers because they have identical magnetic environments in a chiral solvents. They show the same signals. So we need to create additional chiral environments to distinguish. Chiral auxiliaries can provide chiral environment to form diastereomers. They mainly include the chiral derivatization agents, chiral sorting agents in the chiral complex. Addition of chiral auxiliaries allow the enantiomers to be differentiated by NMR. Unlike enantiomers, diastereomers have different physical and chemical properties. They show spatial difference. This is correlates to chirality and enantiomeric purity. These three primary chiral auxiliaries operate through different ways. CDA is a derivatization region. It reacts with enantiomers to generate new diastereomers, while CSA forms to diastereomer complex while non-covalent interactions similar to the behavior of chiral-mental complexes. In addition, mental ions can coordinate with chiral analytes to form the stereomeric complex, allowing stereochemical differentiation through NMR. Here we will illustrate the application of NMR in chiral analysis through the following examples. This is a three-component system. The racemic diode interacts with phenolphthalic acid, and the chiroamide reacts with the formal group. Chiral recognition of diode can be achieved by analyzing the distinguishable NMR signals of protons. Here, chiral with the urea employed as a sorting agent. It forms a diastereomeric complex with analyte through their hydrogen bond interactions. These interactions were stabilized by addition of DMPA through formation of additional hydrogen bond. For chiral-mental complex, the octahedral covalent complex with mental center chirality can coordinate with esters, amines, and ketones to form their stereomeric complex, allowing clear discrimination of proton signals in NMR signals. Traditional NMR-based chirality analysis primarily relies on proton NMR, but its effectiveness is often constrained by signal overlap and limited chemical shift dispersion. So the technology has been developed to simplify the signals. Here, homodecoupling technique simplified quartets into singlets. This facilitates signal assignment, especially for complicated spectra. Over the past decade, fluorinated chiral auxiliaries have attracted attention due to the unique properties of NMR-active fluorine nucleus. Its sensitivity is about 83 that of proton. Fluorine NMR offers a high detection efficiency. Most important, its high sensitivity to subtle changes in the chemical microenvironment makes it advantageous for chiral analysis. In addition, it's broad the chemical safe range, spanning up to 400 ppm, overcomes the limitation of overlapping proton signals. Besides, non-biological background makes it valuable for bioanalytical and pharmaceutical application. Fluorine atom can be incorporated into chiral auxiliaries. It serves as a sensitive NMR reporter for chiral analysis. A large number of fluorinated chiral regions have been developed. For example, we synthesize fluorinated derivatization region. The former group reacts with the amines to form covalent diastereomers. This fluorine atom can sense the chiral amines and the resulting diastereomers show different NMR signals. One signal is R configuration, another is S. This is a chiral-fluorinated mental complex. The fluorine atom is located on the side of mental chiral center. These two fluorine atoms sense the diverse amines. We can select two enantiomers show two separated NMR signals. So, fluorine chiral auxiliaries displayed high effectiveness in chiral recognition. Determining enantiomeric purity or enantiomeric excess is necessary for drug quality control. It's based on their different NMR signals. By comparing the relative signal intensities of the two diastereomers, E-values can be accurately determined. It does not require a calibration curve. For example, in a racemic solution of serine, two diastereomers display distinct signal ratios that correspond to their concentration. The procedure allowed rapid and accurate determination of E-values. It's useful for routine chiral analysis. And in addition, asymmetric reactions are common in drug synthesis. NMR is important for analysis of reaction products. Here, in this sulfur-containing asymmetric reaction, by directly adding a chiral complex to the reaction solution, the chiral product in the solution can be checked and determined. And this result achieves a higher degree of accuracy compared to the traditional HPLC method. This also shows the simplicity and accuracy of NMR measure. In addition, accurate enantiomer quantification in drug formulation ensures chiral integrity and pharmacological consistency. Using an internal standard, the relative integrals of NMR signals enable accurate quantification of each enantiomer. Here, fluorbenzene as an internal standard with a known concentration allows quantification of the diastereomers by calculating the ratio of NMR signals intensities. A key advantage is the convenience of not requiring a calibration curve, unlike fluorescence or HPLC. In addition to chiral analysis, determination of enantiomeric excess values in the quantitative analysis, NMR can also be used for compound structural characterization, analysis of drug impurities, and the study on interaction between drugs and proteins or nucleic acids. NMR spectrum offers valuable stereochemical insights, while its effectiveness depends on several critical factors that introduce challenges and limitations. One major constraint is the sensitivity to chiral auxiliary design. Chiral recognition relies on the formation of diastereomers between the analyte and the chiral region. The spatial arrangement, electronic properties, and the rigidity of the auxiliary are crucial. Subtle changes in structure can alter binding affinity and recognition capability. Fully optimized chiral auxiliary may yield minimal chemical shift difference between enantiomers, influencing both qualitative and quantitative analysis. Another factor is the binding affinity between the auxiliary and the analyte. Weak or non-selective binding can result in poor resolution and ambiguous stereochemical assignment. Lastly, overlapping signals and the resolution issues present practical limitations, especially in complex mixtures or when analyzing analyzed with similar chemical environments. Small chemical shift difference between enantiomers can be obscured by spectral congestion, baseline noise, and multiple overlap, particularly in proton NMR. Even in fluorine NMR, while signal dispersion is broader, careful spectral deconvolution and high-field instrumentation may be necessary to resolve closely spaced weak peaks. These challenges suggest the need for meticulous chiral auxiliary design and advances data processing to harness NMR potential in chiral analysis. So, the development of high sensitivity NMR technology is essential to enhance quantitative capabilities and broaden applications. In addition, to broaden the scope of chiral analysis, it's necessary to develop chiral regions with enhanced solvent compatibility and incorporate alternative NMR active nuclei to expand their applicability across diverse chemical environments. Recent years, the rapid advancement of AI has permeated every scientific field, offering transformative tools for data analysis and prediction. By integrating experimental techniques with computing technologies, efficiency, accuracy, and the throughput of chiral analysis can be enhanced. This also will facilitate the interpretation of complex NMR spectra. In our recent work, we developed a machine learning-based chiral prediction platform using fluorine NMR chemical shifts, addressing a critical gap in enantiomeric discrimination and expanding the capabilities of fluorine chiral auxiliary systems. Thank you, everyone. This is a sharing about the application of NMR in chiral analysis.
不要，你也要补充，还要补充。不要，共享，先别结束嘛，共享结束什么呀？不要。
我下回再。
我刚刚没结束，我只是专机，或者是双机。
那我们要讲到你这个有多余的吗？大概讲一下吧。这是什么？这是视频吗？这是什么？哦，
就是在一维的基础上，我们又发展了二维发展。
这是什么？
就在一个。
这个吗？这我要共享，我怎么共享呢？你
看共享的是不是这个屏幕？
哪个方向？
这是啥？看那个共享的是不是这个屏幕？
刚卡了，你看。
应该是刚刚。
出卡了。要不要？
是不是把它拷贝到这个上面来？
就是卡了，把它拷过来吧。
视频卡了光。
卡了这个把那个关了，你把这个关了，把这个对这个。
对吧？
你播放，
为什么要播放？
哪一个是那个？
不是要讲新的吗？不是要讲新的吗？那就讲吧。好，好，你来弄吧。
放二维那边可以了，一维二。
从2维来2维里面讲啊。
I 按一下。
这个
I introduce the 1D following of 1D following NMR. Now I will add some content to introduce 2D and 3D NMR about following CDA or CSA agent. This one I introduced before. Floating agent was introduced. A floating agent was introduced into a CDA agent, and this chiral floating agent contains the chiral amide. So, in the 1D floating NMR, we consider two distinct floating signals. So, if we introduce two types of floating agents, we consider two types of floating NMR signals, right? So our group developed two floating agents mocking the CDA agents. Like yeah.
这手里是这样的。
Like this region, the triple fluid method and the floating agent was introduced late.
柠檬这个，对这个。
Yeah, like this region, two type of floating group was introduced this compound, and we can see that in a high field region, is this floating agent signal. However, here lower field region shows this one signals. So these two type signals we can remap the NMR signals to 2D. About the three type of floating group into a chiral region, we can see that this three type of floating group show the different different NMR signals, and we map these NMR signals into a 3D. So now our group are developing a 2D or 3D the chiral region to include energetic resolution. So this is our share about 1D, 2D, and 3D floating chiral auxiliaries. Thank you.
黄老师要补充一下。Next line。就是最后有一个讲说。
So we can see a problem. The resolution of 3-dimension problem is highest. So we can in three dimension. We can easy to detect the signal of sound. So I think this analytical method is very interesting. Okay. Next, our member of our group, Dr. Gu Dandan, will introduce chiral HPLC method. Okay, please.
I want to ask one question. What about these chiral compounds inside the body? When you give these compounds, we have enzymes in our GI tract and in liver that can convert these chiral forms. We have enzymes in our body, in intestine and in liver. They have the ability to convert R form into S form. So if you apply these chiral forms inside our body, so they can be converted into the pterogenic or other forms that are harmful to the body. They are just trying to separate when we have a mixture. I was asking about thalidomide. Thalidomide is a drug.
I will discuss after this lecture.
Thalidomide is a drug. Ketamine is a drug. Ethambutol is a drug. That's why I was asking from the professor. His presentation has these drugs, and they are already in practice.
不知道是这是播放的那个视频放完了。
Even from that case, our form is not the same.
这是黄
But
志
we are
放的视频，
thalidomide is in use against
Found
cancer. Ketamine is an anesthetic agent.
这个先不玩啊。
直接显示页面。
ये सारा at the end drugs के
谁的？
不是我的还没打开，要打开我这个是可以，你看现在这个是这个视频给占用了。
ये जिसके question
那咋整？那咋整？
那就是你先显示桌面，然后把我的那个视频给打开，然后再共享。
先显示桌面什么？这不就是桌面吗？
ब्लड का जो ब्लड वो 1 महीने से 6 माह हमारा पे काम है। आपकी इंटेस्टाइन और लिवर में साइटोक्रोम P450 उसको कन्वर्ट कर देता है अगेन इनटू द S फॉर्म और वो टेरोजेनिक
共享。 这个答案。
देखिए इसमें है ना ये प्रोफोर्स पे बता रहे वो ड्रग्स पे बता रहे जो पहली थी ना प्रेजेंटेशन
等一下，再我先说一下。
好。
मैं नहीं
Good afternoon, everyone. I'm Dr. Gu Dandan. It's my honor to share the important topic about chiral analysis based on HPLC. I have prepared a video. Okay.
Hello, everyone. Welcome to today's lecture on chiral analysis based on high-performance liquid chromatography. I'm Dr. Gu Dandan. It is a great honor to share with you this important topic in analytical chemistry. Let's start with a simple question. What is chirality? Chirality is a property where a molecule and its mirror image cannot be superimposed. Just like your left and right hands, they look similar, but they are not the same. You can find chirality everywhere, such as ancient Chinese characters. In nature, we can find chirality in snails and vining plants. We can also find chirality in biological molecules, such as DNA and protein. In small organic molecules like lemony, one form smells like lemon, the other like orange. Another famous example in small molecule is the drug thalidomide. One form helps with morning sickness, but its visual image causes birth defects. This shows why chirality is so important. Now, let's go through some basic concepts. Chirality is defined by the non-superimposable mirror images of molecules, like left and right hands. Molecules with chiral centers exhibit handedness, leading to left hands and right hand forms. The significance of chirality lies in its effects on molecular behavior in medicine, food, and the environment. For example, in the medicine, two mirror image forms of a drug can have completely different effects. One might be helpful, but the other harmful. Apart from biomedicine, chirality also has a significant impact on other fields. In the food industry, chirality affects taste and smell. In environmental science, chiral pollutants can harm ecosystems in different ways. Therefore, the FDA and EMA require the development of a single enantiomer of a drug candidate if the enantiomers differ in their pharmacological action or toxicological profile. So, separating chiral molecules is not just a chemistry problem, it's a matter of safety and effectiveness. Among various analytical methods, high-performance liquid chromatography is one of the most commonly used tools for chiral analysis. Now, let's talk about HPLC. HPLC, high-performance liquid chromatography, is a technique used for separating components of a mixture. It is widely used in analytical chemistry for the identification, quantification, and purification of components. The HPLC system has 5 main parts: injector system, transport system, separation system, detection system, and data processing system. The analyte enters the chromatographic system through the injection valve. After the action of the mobile phase in the transportation system, it is separated by the chromatographic stationary phase in the separation system, and finally undergoes qualitative and quantitative analysis through the detection system and the data processing system. Let's look at a real example. In hair dyes, there are chemicals called phenylenediamine. There are 3 types with different toxicities. Using HPLC, we can separate them quickly and safely. Under specific conditions, like using methanol and water as the eluent, we can detect and measure each type accuracy. Another example, fractoligosaccharide, which are used in functional foods. HPLC helps us separate and analyze these sugars to understand their structure and health benefits. It helps food scientists develop better products. Another example, DMOI is an important active component of vine tea, accounting for about 20 to 40% of the total content. It possesses various effects, such as anti-inflammatory, antioxidants, and blood pressure reduction, and so on. Using HPLC, we can measure how much DMOI in the tea, which helps in quality control. Now, back to chirality. Why do we need to separate chiral molecules? Firstly, it helps to understand their different behaviors. Secondly, it also helps to make drugs safer and more effective. In addition, it is important to improve analytical methods and meet legal and safety standards. Generally, there are several chiral separation and analysis techniques, each with a specific role. First, we have chiral NMR. It is great for identifying which enantiomer is which and checking purity. It is just for analysis. It cannot physically separate them for you. Next, there is chiral spectroscopy. It's a quick way to check if a sample is pure, but again, it doesn't separate the molecules. Then we have chiral crystallization. This is a classic method used in factories to get pure enantiomers in large amounts. It's excellent for separation, but it's not used for precise analysis or quantification. Finally, we have chiral chromatography, like the HPLC we are discussing today. And this is the key point. Chiral chromatography is the only one that does both jobs perfectly. It can separate the enantiomers and at the same time precisely measure and quantify them. This powerful combination of being able to both separate and analyze is why chiral chromatography is the gold standard in modern labs. Now, let's dive into how it works. The chromatographic method for chiral separation and analysis can be classified into three types. They are chiral stationary phase method, chiral mobile phase additive, and pre-column chiral derivatization method. The first one is chiral stationary phase method. One common way to separate chiral molecules is using a chiral stationary phase. This is a special material in the HPLC column that interacts differently with left and right-hand molecules. The chiral selector in the stationary phase binds to one form more strongly, so they will elute out from the chromatographic column at different times. Scientists can design this chiral stationary phase by introducing chiral recognition sites onto the surface of the matrix. For example, we modified catexon to create new selectors that are coated onto silicon gel. Firstly, preparing two catexon shift-based derivatives by introducing two kinds of chiral substitute benzamide groups at the C2 position on catexon. Subsequently, the resultant CBS were modified with two different final carbon mates at the C3 and the C6 position to produce 4 new catexons. Finally, coating the chiral selectors onto the surface of silicon gel matrix. The obtained chiral stationary phases have been used to separate many chiral drugs successfully based on the interactions with the chiral recognition sites. Another method is to add a chiral additive to the mobile phase. Additives like cyclodextrin can bind to chiral molecules and help separate them, even on a regular column. This is a flexible and cost-effective approach. Adjusting the concentration and type of additives, along with pH and temperature in this method, is crucial for achieving optimal chiral resolution. For example, the DMY has 4 chiral forms. By adding beta-cyclodextrin to the mobile phase, we can separate them on a standard column. This is very useful for analyzing natural products like wine tea. The third method is pre-column derivation. Here, we chemically modify the chiral molecules before injection to make them easier to separate. Specifically, we use a chiral reagent to convert enantiomers into diastereomers, which behave differently in HPLC. For example, amino acids can be derivatized with a reagent called FDAA. This allows us to separate D and L forms on a regular column. This method has been widely used in biochemistry and pharmaceutical analysis. Next is a summary and outlook. In conclusion, chirality plays a critical role in pharmaceutical, food, and environmental science, influencing safety and ecological impact. HPLC serves as a powerful tool for chiral separation, enabling precise analysis and purification of enantiomers. Methods such as chiral stationary phase, mobile phase additives, and pre-column derivatization effectively address diverse chiral separation challenges. Continued advancement in chiral HPLC techniques will further support drug development, quality control, and regulatory compliance. However, there are still some challenges. The first question is high cost. High-performance chiral columns and dedicated chiral reagents are expensive, increasing analysis costs. In addition, screening from the suitable chiral column or additive remains a time-consuming, trial-and-error empirical process. Moreover, complex components in real-world samples can interfere with the chiral separation. But the combination with artificial intelligence and new materials may become a new direction for the future development of chiral analysis. We can use AR and molecular modeling to predict separation conditions, significantly shortening method development time. In addition, developing new power materials like morphs and curves as chiral stationary phases may offer higher selectivity and stability. Thank you for your attention. I hope this lecture helped you understand the basics of chiral analysis using HPLC. Thank you. Good afternoon.
All of the examples in the presentation is the work from our group. If you have some work about chiral HPLC, welcome to cooperation with us. Thank you. Which method separating you are looking via HPLC? So I have described three methods. Separate the style molecules. Which method you are using? Are you using or please repeat?
Okay, it's okay. We can hear you.
The first method is chiral also the second we can use some compound models into the chip, but the chiral activity combined. It's the professor of work. So currently, have you published any method that has separate chiral molecules? All of the works that published, and applied in many different samples. Thank you.
After purification, where you applied these chiral compounds?
Sorry.
After purification of the chiral compounds, where you applied them? For what purpose you are purifying them?
You mean where we use the column?
Yes. Column chiral compounds that you purified. Chiral compounds.
Purified. Actually, maybe we balance analysis in our.
Okay, and outside the body or inside the body?
Outside the body.
Outside, in vitro.
Outside the body.
In vitro analysis. Thanks.
Okay.
Does you synthesize the chiral molecule? Does you synthesize the chiral molecule?
Yeah. Actually, it was inside by our students.
I curious, how many stereogenic center have you been success for the chiral molecule? Have you been success?
That's how
in one molecule, number of chiral centers can generate, we can synthesize. In one molecule, how many chiral centers can generate?
maybe we must one center. We also have multiple ones. That's like
Okay, any question about another analytical methods? Kind technology. So our group have developed many kind of analytical methods, such as kind of HPLC, kind of and cycler ECD, and to detect many kind of couplers in natural extracts, and in order to detect the change of kind of compounds. So I think chirality is very interesting and very important in for many research fields, especially for TCM, Chinese traditional medicine, or let's say natural herb.
Given in our body, chirality is very important. If it's L amino acid, that is the part of protein. If it is D amino acid, then it works as a neurotransmitter or as a regulator of hormones. We also have D amino acids, but for different functions. For our immunity, we have D amino acids, but that are not the part of protein. They function individually. So chirality is also important in our system. So in future, you are planning to use these chiral compounds for in vivo applications?
No.
To use that? In future, you are planning to use these diagnostic probes as a diagnostic probes to detect cancer inside the body.
Start. Sorry. I think many D amino acids might be is extraneous in body, and many D amino acids are changed from the air amino acids in body. So there are many environmental factors which can influence this conversion. So maybe we think the amino acids are harmful to harmful.
Yeah.
Yes, harmful to people's health, but
they are not.
Yeah, yes,
They are
maybe not. Maybe not.
They are also the part of our body.
Yes.
The amino acids also has function in human body.
Yes, maybe they are have functions.
Different than the protein synthesis, but they have different functions.
Okay, yes, but there are few research about the function of the amino acids.
Yes.
So there are I remember there is international meeting about the amino acids. The first meeting is opened in 1990 in Tokyo. This meeting is I think we can join this this meeting. Sorry.
Thank you.
So maybe we can cooperate in this interesting field. Okay.
Yes.
Thank you for your attention. Okay.
तस्वीर बना है तस्वीर के लिए तो उठेंगे बनवा लेते हैं
Picture.
Yeah.
So picture, group photo.
Group photo. Oh, yeah. Oh, picture. Take a photo. Yeah. Oh, maybe. 那我那是。
要拍个合影。
那个学生那个给我们拍一张照片。
Ask them you will. Slideshow first page. Oh, okay.
Take a photo together.
Yes.
The slide and certificate. Where is certificate? Yeah. And certificate.
Show his slide. This professor's slide.
Yeah.
Students
The first
click
professor
one.
slide I think is
One.
better.